Hello everyone. My name is Ashutosh Kumar and I work as a professor in the Department of Bioscience Bioengineering, IIT Bombay. I am a structural biologist, use NMR as a primary tool to elucidate structure, dynamics, interaction of proteins. Recently, we are looking at some of the biologics and biosimilar product. So, in this lecture, what I am going to do to show you or show an exposition, expose you how NMR spectroscopy can be used for robust assessment of higher order structure of biologics. Higher order structure means structure which is like there are 4 levels of structure in proteins. So, primary, secondary, tertiary and quaternary structure. So, higher order structure includes the secondary structure, tertiary structure and quaternary structure. So, in this lecture we are going to discuss how NMR spectroscopy can be used for elucidation of higher order structure which includes secondary, tertiary and quaternary structure of protein. So, over the year actually X-ray crystallography has predominantly used as a structural tool and actually it has dominated the structural biology field for many years. Because of its fast speed, here you just have to get a crystal of a protein, and if this crystal of protein diffracts well, you get a diffraction pattern and you can solve the 3-dimensional structure of a protein. So, using these structures, basically scientists develop a paradigm which is called a structure function paradigm. So, you know the structure, you can learn a lot about a function of a protein. But actually to understand completely the structure of a protein which comes in 3 dimensions, you see there is another dimension which is very important, and that is a 4th dimension of the protein structure. Because protein are not a static entity, they are dynamic, they have a motion. And these motions are at different time scale all the way ranging from picosecond to second time scale. Now, to understand completely the function of a protein, this 4th dimension which is time domain data, time dimension is required, and therefore 3-dimensional structure requires a 4th dimension. It is simple like that. So, 3-dimensional structure is like necklace, but the necklace is not beautiful in itself. When it decorates a beautiful neck, it becomes much more relevant. Similarly, if you have to understand the protein function, actually you require the time domain data to decorate it and make it more functional. Therefore, over the years this structure function paradigm has been revised. Now, to understand the function of a protein, you need to include the how protein folds, what are the folding pattern, what are the folded portion, unfolded portion in a protein, and then what are the associated dynamics of a protein. So, structure together with folding and dynamics happening at various time scale together constitute a function that will be performed by a protein. It's something like that. So, if you solve a structure mostly by crystallography, you see something like a static frog, but frog is never like all the time, it is not static, it has subtle dynamics. And when it does dynamics, like putting out its tongue to catch a fly, but that happens for a fraction of a second, it puts out its tongues, but that putting out tongue is very much important for doing dynamics or doing function of a frog. So, you require a tool which can club this structure with subtle time scale motion that is done, or certain motion that is done by this frog to understand completely behavior of a frog, and therefore 3-dimensional structure requires a 4th dimension which is time dimension. Now, in this aspect, NMR has emerged as one of the prominent tools. So, you see why NMR solves this problem, because it gives from the same sample structure and the dynamics information. Now, the famous person, Nobel laureate Professor Richard Ernst, who got the Nobel Prize for developing Fourier transform NMR in 1991, once he has said, because there is hardly any technique that is so informative for so many different type of application, and because no other technique provides so much fun. Now NMR not only gives you information, it's doing the experiment with NMR, it's also a pleasure, because every time, every day you learn new things. So let's go back a little bit of theory of NMR, why and how NMR evolved and what can be investigated using NMR. So see, NMR is nuclear magnetic resonance. Here we are exploiting the nuclear property of an atom to understand its property. Now if you look at many of these molecules have some or other nuclei which is amenable for NMR, like various state of the samples like proteoliposome, powder, frozen solution, microcrystalline, hydrogel, precipitate, aggregates or biopolymer. All of these has protons, or all of these has some or other active nuclei which can be exploited in NMR spectroscopy. Now coming back to the interest of today's topic, which is protein. So when we look at structural biology, we exploit that different nuclei present in a protein, something like a proton which is naturally abundant. But if we want to do multidimensional experiment, we isotopically label these proteins with something called N15 or 13C. So what we do? We feed our bacteria with 13C level glucose and N15 ammonium chloride, or even we grow the tissue or cell in this labeled media. So when they produce protein, that will be labeled. So we can work with unlabeled protein or labeled protein. We isolate and purify protein and check their purity in one of the classical technique by doing SDS page. Single band determines the high protein quality, or one can do mass spectroscopy like MALDI. If you have a pure protein, you put that in NMR tube, put it in the magnet, record various correlation data, like here I show you 2D homonuclear correlation TOXI and NOGI, and or you can even record a heteronuclear data. So combining all those data, we do resonance assignment, and using all those information, essentially we solve the structure of protein or peptide, and you see here ensemble of protein structure. Now NMR always gives you ensemble of a structure rather than a unique structure, and this ensemble structure has also the dynamics information hidden here. So if you look at this portion, essentially is quite well conserved, and this portion has flexibility. So this is in nutshell 5 step what we do when we determine the structure of a protein using NMR spectroscopy. First is sample preparation, where you can isotopically enrich, or at natural abundance one can also work. Then record data for weeks, then assign all those, do the resonance assignments, and then using the angular constraints and distance constraint, you solve the structure of a protein. So I was doing typically classical biology. I was working on a small protein peptide called amylin, or it's called also human islet amyloid peptide. It is in interplay with insulin, so it's co-secreted from pancreatic beta cell, and it is responsible for satiety and like slowing down the emptiness of a stomach. Now it is co-secreted with insulin in ratio of 1 to 2 to 50. But what happens basically in case of type 2 diabetes, when insulin is secreted more in amount, this also gets secreted, and then it starts aggregating, forms the amyloids, which ruptures your insulin producing beta cell. So that becomes dangerous. So I was working on this peptide which is like co-secreted with insulin, and we are doing structural biology of this. So we were trying to elucidate the structure of these fibrils. We are trying to look at the toxicity. So here you can look at HIPP have more toxicity than the normal control, and it forms fiber-like structure. We also look at some of these, how can we disrupt these fibrils using a small molecule. So this is meracitin, which is found in jamun or any citrus compound, and they binds with the fibril, disrupt this fibril. So we are doing this, and then I have a friend who works in Biocon. So we were talking, and you know, many times talking with people with different background is always useful. So he said, anyhow we were working on amylin, why don't you also start looking at insulin? It's similar, like slightly bigger in size, but it has lots of interesting questions hidden in that. So now I started looking at that, and we were working on amylin, which aggregates. There is another amylin from rat where there are some critical mutation, and in the rat amylin does not aggregate. Now taking a clues from here, people designed an amylin analog or a variant of amylin which is called palmitide. Now this is used in like as an anti-diabetic medication. So if you look at here clearly, what people have done, change some of these serine to proline, and that's how it does not aggregate, and this is sold as a drug. Now classically we are doing this and we encountered this. So we thought that the biopharmaceutical can also be an important target to look at, and that's what I initiated I got initiated in biopharma research. So when we started looking more deeper into that, we found that market of biosimilars are growing. The biological drugs are a drug for future. If you look at in last 4-5 years, it has increased exponentially, and it is thought to be increasing quite rapidly in coming years. So, and as you go in increasing, the more and more molecule will come in biosimilar field. Their characterization with a robust tool is required. So if you look at 2018 to 2025, here in 5 years it seems to be tripling. And all over the world, whether it is America, Europe or Latin America or Asia, everywhere it is increasing. Now, so how this biosimilarity development program works? So biosimilarity, essentially you are making a drug of already approved biologics drug, and this drug has to be similar in all aspects. So it is based on totality of evidence. So when one is doing this biosimilar manufacture, so if you look at the innovator path, in innovator path a new molecule is being launched or a new biologics drug is launched, lots of clinical study is required, then slightly less clinical pharmacology, then preclinical, and then analytical. But when we are making a biosimilar of this innovator molecule, you have to first match in all physiochemical property these innovator molecules, the biosimilar molecule with is matching with the innovator molecule. Therefore, robust analytical characterization is of paramount importance. So you do very like a thorough analytical analysis, then you do preclinical, some clinical like a less clinical pharmacology, PK/PD, and finally the clinical study, and that is conducted only on the sensitive population with sensitive endpoint to design or detect a difference if there is one. So in this case it is an inverted pyramid, that means robustly analytical characterization needs to be done if we are going for a biosimilar like a production or manufacturing program. Now typically there are various techniques or methods that are used for characterization of biologics. Right, there are analytical methods which are used for characterizing a small peptide or protein. First you have to make sure that your primary sequence is exact, so you do mass spec based analysis, fragmentation, and do match the fragments which matches with the reference RLD product. Then one has to do physiochemical property, like if there is a modification, there has been become some introduced modification or PTMs, glycation, phosphorylation or etc. So do physiochemical analysis or charge variant analysis to understand everything of that. So if after doing this physiochemical analysis, it is important to understand if the protein is folded in a correct form. Now correct form folding is a domain of a structural biology, and that's what I introduced to you. I am a structural biologist, so I work in this domain looking at the higher order structure of biologics and biosimilar, where we will look at the secondary, tertiary, quaternary structure of the protein. So once we establish the correctness of the folding of a biologics drug, next question one has to address, is there any oligomeric species or aggregation state of the biologics drug? During the folding, during the formulation, have you introduced any aggregation state? Now these aggregations are very dangerous, so it has to be robustly established that we are not introducing any aggregation state. After that, it's impurity analysis. Impurity can be introduced from anywhere, whether it is manufacturing related or during the process some degradation happened. So one has to really look at this. So here again some of the orthogonal techniques like AUC is very important for oligomeric state analysis or impurity analysis. Finally, you do biological evaluation. So you will state the mechanism of action, whether this drug is binding to correct receptor, what is the response after the receptor, that has to be done. Now, the impurity analysis is very important because if there is an impurity, it causes immunogenic response. So therefore, together with correct folded structure, your oligomeric state analysis or aggregation analysis is very important. Now, typically methods that are used for higher order structural analysis are follow. There are many low resolution techniques, something like chromatographic technique. So what you do, you purify protein, run on size exclusion chromatography, helix exchange chromatography, or ion chromatography, all of these, and see what is the retention time, both the RLD and your biosimilar product is eluting at the same time or not. So this is one of the techniques to establish. Then there are some low resolution structural techniques like a circular dichroism that gives overall idea about secondary and tertiary structure. Then FTIR or Raman, it works on vibrational spectroscopy angle, and also it gives idea about secondary structure. Then fluorescence is also used to establish kind of a folded structure or tertiary structure of a biologics drug. Then looking at the aggregation or molecular state, light scattering or dynamic light scattering is used. But along with these low resolution technique, there are some high resolution technique like hydrogen deuterium exchange with mass spectroscopy or NMR spectroscopy are used. X-ray crystallography that we started with, they are also used. So you have to crystallize the proteins, biologics or biosimilar, and compare how the diffraction pattern or structure is coming. Now slowly cryo-EM is also becoming popular for big big biological molecules. So these are high resolution techniques that are used. Now, so you see biologics are large, and protein based therapeutics actually vary from size and complexity. If you take a small molecule of 180 kilodalton like aspirin, this is very small. But as we enter into biologics regime like insulin, it's a like 5,700 dalton, it's quite a big compared to this. If you go even further like a growth hormone, it's a 22,000 dalton, and monoclonal antibody 150 kilodalton. So that's quite big molecule. Now if you compare with aspirin, the complexity increases, therefore robustness of the characterization is needed when we are talking about the higher order structure. So you see the little bit variation that happens between this structure and some other structure needs to be captured in a robust and precise manner, and there actually NMR becomes exciting tool. Why exciting? Because first of all, you don't perturb molecule, right? So biologic drugs are complex macromolecule, and using two-dimensional NMR, you actually separate the signals. Now instead of one signal or two signals, you have many signals to compare. So if there is a deviation, that you can easily capture, right? So and also, since I showed you that it is used for a structure determination of a biomolecule, it provides an opportunity to assign these signals and see if this variation is coming from where. So one can address those and correct it if needed. Okay. This other major advantage of NMR, it's a highly reproducible. If you do experiment with all correct parameters n times, your data will be reproducible n times. So you are very confident that every time I do experiment correctly with a same sample, if sample is not changing, a spectra will be always reproducible, right? So that's a good advantage. So therefore, it can be implemented on different biologics with a different batch and composition. You can do batch to batch analysis to show the robustness of your production. The only challenge that we have in NMR, It is one of the insensitive technique, therefore, it require typically high concentration of the biologic drug, right. So, that is one of the limitation. Otherwise, in terms of reproducibility, in terms of wealth of information, it is profound. Now, let us take some of the example of small drug molecules like insulin or liraglutide, and I am going to show you how NMR can be used for robust structural analysis. So, if you look at insulin, it's a like a 5.7 kilodalton. It has two chain, A chain and B chain. There are 3 disulfide, and it has like a helical structure with a small beta sheet. Insulin oscillates between this monomeric structure and hexameric structure. Most of the time, when in the formulated state, it is stabilized by this zinc and potassium sulfate. It is a hexameric structure. The other molecule that I am going to little bit show you about liraglutide. This is also anti-diabetic drug, GLP-1 analog. So, these are the two molecules that we are going to talk. It is a small peptide based drug. Let us start with that, right. So, the one can do the 1D NMR, just proton based NMR. You record your IHDP in-house drug product with RLD1, RLD2 and API, and compare resonance by resonance. If you compare here resonance by resonance, one thing you can see that the peptide structure in the formulated state is same. Lines are generally sharp here, so one can see if peptide is is in monomeric state. So, just by recording a simple 1D spectrum, we are saying it. So, if you now zoom little bit more, like let us look at RLD1 and RLD2, there are some minute difference you can see here. So, that one can see this slight difference is there, and that slight difference because of buffer composition or something like that, right. But overall spectral profile of your biosimilar drug with RLD is matching. So, even it little bit variation, like if you look at here, little bit variation between these, you can capture it. Now, that is a qualitative, looking at just by recording 1D spectrum, we looked at qualitatively that peptide structure in the formulated state is same. Even between two RLD there is minute variation, and your drug product is overall matching with the RLD. That is what information we got, but that is not enough when we are doing NMR. We can go beyond this. Now we do the 2D spectrum, where we establish the proton-proton 2D correlation of a peptide drug. So, there are very simple experiment, something called TOXI, total correlation spectroscopy, and NOSI, which is called like a through space nuclear overhauser effect spectroscopy. This is a through space spectroscopy, this is through bond spectroscopy. So, this NOSI gives essentially about the correct folding pattern or so. One can record it, and say on insulin we can record here TOXI and NOSI. Typically with like a formulated condition, it takes about 13 hours to record, and then you have a beautiful spectrum, well dispersed peak. So, clearly says that now peptide is well folded. Looking at this NOSI pattern, lots of peaks are there. Now, that establish the protein is folded, you can even solve the structure using this, but probably in biosimilarity you do not need to solve it. And here remember, we are doing at natural abundance, we are not feeding anything to the growth media so that it is labeled. So, we are doing at natural abundance. Now, if we compare two NOSI, and one can establish the correlation between those two, but before that we can also establish some critical quality attribute of this peptide drug. So, remember I showed you that there are 3 disulfide bond in insulin, and looking at the NOSI spectrum, you can easily show this NOSI connection between say A6 to A11, B19 to A20, and this is the way you can show that how disulfide bond is forming. That is a fantastic, right. This is one of the few techniques that show you how after folding the peptides are coming closer, and then you can do this NOSI spectrum to show the correlation. So, you can show the disulfide bondage has formed correctly. Now, this is one of the critical quality attribute of insulin correct folding. Now, one can now compare the two drug formulation, and you can see many of the peaks are matching, but some peaks are even not matching. So this is doing simple Toxi and Noji, you can show the degree of similarity that is coming between these two experiments. Now if your molecule is bigger, so many times you have to label the protein. Typically in biosimilarity that is not preferred, but if you want, you can go for a natural abundance HSQC experiment, right? Natural abundance, you see the N15 is 0.037% populated at natural abundance. So you can record a longer experiment between, say, this is filgrastim, one of the drug used for the cancer. So you can use this originator and biosimilar filgrastim and show the residue wise similarity between these two products. So here each dot represents one amino acid, and in a residue specific manner, in a quite robust manner, we can show the biosimilarity between the biosimilar drug and originator drug. But this takes longer time to record, because this is we are recording at a natural abundance. There is another important group present in the protein. So if you look at methyl groups, now methyl has a nice property, it has 3 protons, and these 3 protons like they oscillates like in a rotameric motion. Because of that, it has a favorable relaxation property, and there are 6 amino acids in a protein that have a methyl: alanine, valine, leucine, isoleucine, threonine, and methionine. And if you plot those methyl containing amino acids, they are distributed all over the protein. So if you capture these methyls, we can again using this simple experiment establish the higher order structure similarity. By capturing this analysis, I showed you it has a favorable relaxation property, it is distributed all over the protein. So if you capture only methyl in a short-term time, experimental time, we can establish the similarity between these two products in less expensive manner. So that's what has been done here. It's a experimental time is less than 2 hours compared to 13 hours that I showed you for Toxi and Noji. Now here you can see beautiful spectrum emerging from methyls of insulin, and you can even count all the methyls, you can annotate them, assign that these are, say, coming from isoleucines, and so forth. You can assign there and find it out structure as well as dynamics happening in this protein. So here is the assignment, you can assign it. It requires, of course, help of Toxi and Noji and other experiment, but now after one time we have assigned it, we learn about each and every amino acid here. Great. So little bit going into before, now we have a tool how we can study the insulin in detail. So now let me bring you to, or let me walk through little bit of dynamics insulin plays. So generally insulin are formulated in 3 forms because of different dose requirement by different person. So insulin is the first personalized therapy, right? Some person required rapid release of insulin. So for them it's called insulin R, regular insulin or natural insulin, or rapid acting insulin there. So it starts working in 30 minutes and lasts for 5 to 8 hours. Some people who needs quicker insulin in their blood. There is another formulation which is called insulin N. So it's NPH, long or intermediate acting insulin. So this comes in a suspension form. It starts working in 1 to 3 hours, approximately lasts for 10 to 18 hours, and this is stabilized by zinc and protonium sulfate that shift equilibrium towards crystalline state of a protein. The another kind of people will require pre-mixed insulin, where they require regular insulin mixed with this N insulin. So here it's called 70-30, short acting insulin and fast acting insulin. Now why is so? Because this is the blood barrier. Insulin, when it is in hexameric, it has a minimal diffusion, but when it is in monomer, it can cross the blood-brain barrier. So now if you can formulate in a hexameric state, slowly it diffuses in a relaxed, like there are 2 forms, tensed and relaxed. Slowly it goes to relaxed one, then to dimeric, and then to monomeric, which goes into the blood to activate, bind with insulin receptor, activate the GLUT4 receptor, glucose is coming. Now this equilibrium is very important. In some person you need rapid, in some person you need slow. So therefore these are mixed. So when you are mixing two kind of biphasic, a crystalline form and a soluble form, you need a technique which can investigate it. And that's what our NMR helps you. So if you record an insulin N or insulin R, you see these kind of a spectrum. So this is actually, here we have shifted the equilibrium towards monomeric by putting acid here. We brought it to pH 2.3, but in those cases also you can see crystalline to soluble form, there is some difference in the spectrum. Now you can match it with insulin R and 70-30, you see there are peaks are shifting. So that says that there is finite difference in the monomeric state that we have forced by putting acid in these two cases, insulin R and insulin 70-30. Overall insulin is same, but intricate structure is slightly different. That is what our NMR data is saying it. Now that is all about a little bit about insulin. Now one problem that often we have in doing proton based NMR spectrum, because we have excipients which is high in concentration. Now this excipients many times comes at the region where we have a methyl peaks. So this is kind of a headache. We want to get rid of these excipients. So the people in the field develop a filter called Sierra filter. If you invoke this filter in your pulse sequence of methyl-TROG that we are discussing, it cleans up the excipient peak signal like selectively. You can see excipient interference coming from liraglutide. Here you have a little clumsy spectrum. If you put Sierra filter, you can have a beautiful spectrum. So each of these dots represent one methyl group, and you can study a lot using these methyl groups, right. So that is all qualitative. Can we go quantitative? So Bruker with Mestronova developed a tool called MNOVA BioHOS. So you can record the spectrum like RLD and IHDP here, IHDP2 and RLD, and overlay the peak, and it does peak picking, looking at the intensity, looking at the chemical shift, and it plots the correlation coefficient. So now if it is matching very well, you get a very high correlation coefficient like here 97%. If there are some differences, you can see qualitatively, not visually that much, but if there are differences, you get a poor correlation coefficient like 88%. So not only qualitatively, but we can also establish quantitatively how similar or how different they are based on the spectrum that I recorded. This is all methyl HSQC that I was discussing you of RLD and IHDP. But there are many other methods for doing a statistical analysis. Other one is called CCSD, combined chemical shift deviation, which takes the proton chemical shift, carbon chemical shift and intensity and plot in this equation and show that how two peaks are similar and dissimilar. So for the previous one can see the RLD1 versus IHDP, RLD2 versus IHDP, RLD3 versus IHDP. So here all the peaks are matching very well. So deviation is very less compared to this. This is like lots of deviation, here slightly less deviation. So depending upon how much similarity and dissimilarity we have in a quantitative manner, now we can approach it. Now suppose if the spectrum is not that well resolved that we had in the methyl CoG, if spectrum is not very well resolved like here or like here, then you can just record a NOG spectrum and of two product RLD and IHDP and take a difference between these two images, which is called image difference analysis, and you can see where are the differences coming. So you can that also you can pick up. So this is another method for understanding the similarity between the biosimilar versus RLD. Great. So that's all about structural information. Can we go slightly deeper, because I told you aggregation is also very important. So for aggregation analysis, along with other technique, DOGE has emerged, DOGE NMR has emerged as one of the prominent tool. DOGE called diffusion order spectroscopy. So basically it measures the translational diffusion that depends upon mobility of the compound, and in a mixture of various compounds they will have a different diffusion coefficient, and one can investigate that using Doji NMR. So, even if suppose a small molecule binding to a large protein, you can get it. Aggregation kinetics of a molecule, you can find it out using Doji. It is simple based on the gradient concept of the NMR. So, here suppose here are two molecules, they experience different gradient, and then you give a time delay, then they will be diffusing differently, and then you reverse the gradient, they will be diffusing. So, depending upon how they diffuse in a solution, in influence of these inhomogeneous magnetic field introduced by gradient, you can find the translational diffusion of this molecule. So, you can find the decay of the intensity with the gradient strength, you plot it, you find a d value. From d value diffusion coefficient, one can calculate the r value hydrodynamic radii using this Stokes-Einstein relation. Now, that is what we did for one of the insulin analog API, and you see the theoretical diameter is 3.2 nanometer. When we measure with a Doji, we are getting the hydrodynamic radii is 1.75, that is quite closely matching. So, then we went ahead and did diffusion coefficient analysis and RH for like many other, here diffusion coefficient analysis for many other insulin variant, and you can see more or less they are falling in this region. So, for this API was 1.58 and many formulated concentration coming between 1 to 2. So, shows that predominantly they are in the state that they should be, and that is what we are getting the nice diffusion coefficient for each of these molecules. Now, another important parameter, why structure is important, why you need to know whether my biosimilar is of having good structure. One of the example I am going to show the structure function correlation of insulin. So, here you see human insulin, very nice spectrum. Now, we combine this with SPR, surface plasmon resonance, that measures the binding of this insulin with a receptor. Here IGFR is a receptor. So, we pass the different concentration of insulin on immobilized IGFR, and one can see the response unit in SPR. Using this response unit, you can plot it and find the concentration, the binding constant of the insulin with IGFR, which comes out to be 1.04 into 10 to the power minus 6. It is a micromolar binding. Now, we took another not so nice looking the insulin. You can see this was a nice looking, and red one is not so nice looking, lines are really broad, and if you show the binding, it is a 17 micromolar, 1 versus 17. So, if your protein is not folded well, its structure is not nice, you see you are not going to get good response. So, therefore, it is recommended that you work robustly on getting the correct folded form of insulin before you really go for a functional analysis. Now, that is what we show, that structure, getting the higher order structure is extremely crucial before we really go for a functional analysis of these biomolecules. So far so good. So this is the next step. We had, so far I have shown you small peptide based drug. Now, we want to go more, what else, what we can look at. Now, you see the another class of drugs that are becoming really popular are MABs, monoclonal antibody. Here adalimumab like a Humira or trastuzumab, they are there. So, now again these are really big molecule, and you know the really big molecule have lots of protons there. So, their spectrum, if you compare, is going to be very challenging. So, now we develop a tool using NMR and using NMR 1D and 2D to show again biosimilarity. So, here this we published in Journal of Pharmaceutical JPBA, Pharmaceutical and Bioanalytical Analysis. So, here originator with biosimilar, we combine two techniques like here DLS and diffusion that I showed you. So, here these two techniques shows the similarity between biosimilar and originator. So, we combined orthogonal technique DLS to show the biosimilarity between these two. So, why, like I was discussing with you, the explaining or doing the experiment with these big molecules is difficult. First, because they tumble very slow in solution. So, their signal decays quite fast. Therefore, it has a poor signal to noise ratio, as the number of signal increases, there is a severe overlap. So if you look at the protein that we are talking about, like insulin or even less than that, 8 kD, tumbling is 4 nanosecond, 8 kD, you can see sharp lines. But as we increase to 16 kilodalton, 24 kilodalton, 50 kilodalton, lines are becoming quite broad. The tumbling is becoming like a tumbling time increases. So if you think about 150 kilodalton, that's even broader. Now working with MAB is challenging, but again here we have a beautiful probes located at different amino acids, these methyl groups. So if you just plot the methyl groups in any MAB, they are presented all over the biomolecules. So FB region, Fab region, FC region, connecting region, all regions they are present. So if we probe these methyl groups, we can really probe a MAB, right? In a, like here we don't have one probe, we have hundreds of probes to look at the critical quality attributes of these biomolecules, and one can exploit it. The only thing you have to do, increase the temperature so that their tumbling increases. Without even denaturing, you can really acquire the spectrum, and that's what we have acquired on our machine. Here you can see, just taking one of these biosimilar that we were discussing in the previous slide, one can acquire the methyl HSQC spectrum at natural abundance in few hours, and this is the well overlapped spectrum of a MAB. So now you can see, you can count easily 80 to 90 peaks are there, and those are coming from different methyl groups here. Right, so one can go further detail and find it out how many alanines, leucine, methionine, threonine, valine, isoleucine are there, and you can do their amino acid type tentative assignments here. So if say something is changing, say methionine getting oxidized or some change happening, peaks are shifting in this region, you can certainly find it out there's something happening in methionine, and you will ask your formulation guy, your production guy to look at the methionine oxidation or combined with a mass spectroscopy to understand this in detail. So one can see with this profound information, now even we can tackle the future drug or a bigger drug called monoclonal antibody using NMR. Now you can again establish a biosimilarity, something like this. So biosimilar and innovator, you can plot the correlation coefficient again using proton and carbon chemical shift and show the correlation between these two products, right? So with this, I have come to the end of this short lecture that we have, how NMR can be used. So towards end, I would like to convey this message. NMR spectroscopy is an excellent tool for pharmaceutical application. You can do lots of things like a ligand based, target based experiments. You can find an epitope that I did not show today, but you can find an epitope from the protein. You can find a binding pocket using NMR. Today what I showed, it is a robust tool for demonstrating the biosimilarity, higher order structural characterization, and not only small peptide based drug. Really we can use NMR spectroscopy for a bigger bio drug up to 200 kilodalton and doing a robust biosimilarity analysis in this case as well. So NMR is a tool for future for biosimilarity assessment. It requires lots of involvement and lots of experiment to be done. Lot more people should be doing this as a preferred techniques. So I think some companies should be early adopter of this technique for showing the biosimilarity. That's what my hope. And with this, I would like to thank the people who has worked. So the funding for doing research comes from Indian government agency like CSIR, DST, DBT, and this spectrometer that we have in IIT Bombay, 750 megahertz Bruker machine that is funded by IIT Bombay IRCC. And in the beginning I mentioned that we have a collaborator from the industry, Dr. Naratna Bajpayee, who works in Biocon. He is heading a division there. So we collaborate on insulin research, and my student Lakshraj work on, he initiated actually with Pramod, he initiated work on insulin and liraglutide, and Jaishankar Singh, who now after completing his PhD moved to Biocon. He was instrumental behind doing SPR experiment. And with this, I would like to thank you for your attention. Thank you very much. All the best.